Welcome to all, and now we will solve another question. Now we will see the question. In a Mendelian dihybrid cross, if one trait shows incomplete dominance, one trait here is the incomplete dominance, and the other trait shows the homozygous dominant lethality. That means when the organism which is in the homozygous dominant lethality, it will die. Then what will be the phenotypic ratio of progenies obtained in F2 generation? Options given here. Now we will go to the solution. Just you can see the first thing, incomplete dominance. If you go for incomplete dominance, as you know here, instead of recessive and dominant expression or phenotype, we will get another phenotype here. Example, we take the allele capital R. When this is the condition, as you know, it gives a red color phenotype. When capital R, small r, now this is incomplete dominance, where we will get another color, pink color. And if you see small r, we'll write here white color. In case of normal dominant, what happens? The plant or organism should get a dominant color than the recessive trait. But here, instead of dominant and recessive, getting another one, that is pink. Remember this, and you will see the lethality. The lethality, homozygous dominant lethality. Now we will take the for this allele capital L. Now these two alleles in the gene are dominant, so this is homozygous dominant, so organism will die in this condition. And another condition, you see heterozygous dominant, capital L, small l. It will give you phenotype, one type of phenotype, and another condition, small l, give you another type of phenotype 2. Indicates phenotype 2 and here phenotype 1. Remember that. Now we will go to the question. We will take these two traits for the dihybrid cross. When you take the F1 progeny self-cross, then the result you will get the F2 progeny. What is the F1 genotype according to Mendel? Double heterozygous. Capital R, capital L, small l. This is double heterozygous in condition according to the Mendel. Now when we do the self-cross between F1 generations, then we will get the progeny of F2. Now we will see gametes for this. When you take two F1 progenies, capital R, capital L, small l. Now this is the F1 progeny, both are F1 progenies. This is a F1 and another organism. Now what are the different types of gametes it will produce? Capital R and the capital L and the capital R and the small l. Right? And this small r, capital L and the small r, small l. And these are the four different gametes from this particular organism. And now see, in this case, the same organism with the same genotype gives same type of gametes, capital R, capital L, capital R, small l, and small r, capital L, and small r, small l. Now these are the four different types of gametes from this organism, and these are another four from this organism. So these gametes all are similar from the F1 progeny. Now we have to do the self-cross between these two organisms, then we will get the results for this question. Now we will analyze this. You will see that Punnett square board by drawing all these gametes. In one side you can take the male gametes, capital R, capital L, and capital R, small l, and small r, capital L, and small r, small l. These are four gametes from the one, and another four gametes same, capital R, capital L, capital R, small l, and small r, capital L, small r, small l. Now we will draw the Punnett square board for this. Right? Now we can see the what are the different types of phenotypes we will get here. As you know, in this case I can take male and the females. When there is a fusion of these two gametes, you get here capital R, and the capital L, capital L. Remember one thing, wherever we are getting for the lethality both are dominant alleles, in that case it will die. That organism will die. Nothing wrong. And capital R, and capital L, small l, and capital R, small r, capital L, and capital R, small r, capital L, small l. And see, when this gamete fits with all the four, capital R, capital L, small l, and capital R, and small l, small l. Now see, when these two, capital R, small r, and capital L, small l, when it fuses with this, capital R, and small l, small l. And you go for third gamete with all the four combinations, capital R, small r, capital L, again capital R, capital L, small l, and small r, both are capitals, capital L. Now again here small r, and one capital L and the small l. Take the fourth gamete with all the four different gametes, capital R, capital L, small l, and capital R, small l, small r, small r, capital L, small l. Now small r, small l, small l. So when you are referring small l, that is in recessive condition, and capital is dominant condition. Now see here the first genotype and what kind of phenotype it produces. I forgot about this capital R. In generally, as we have discussed previously, it gives the red phenotype plus homozygous dominant. It means this organism will die. It will die because of these two alleles are in dominant condition. So next you will see this, or count how many are there like this homozygous dominant alleles for the lethality. Now here one, and you see in this case it is actually pink if it is live and homozygous dominant. So again it will not be live, it will die because of these two dominant alleles for the lethality. And you can find another one here. Again this pink plus homozygous dominant, it will also die. And you see here another organism which is having white plus homozygous dominant, it will also die. Now total among these 16, total 4 will be dying. Now remaining will be there, only 16 minus 4, only 12 will be the live. Now we'll see what are those 12. Take this genotype, capital R, red and heterozygous dominant. I'll take here capital R, and capital L, small l. Now this is actually red plus heterodominant. Now how many total organisms with this phenotype? Now see, capital R, here another one, capital R, capital L, small l. Now red plus heterozygous dominant. It will not die. So total how many are there? 2. Nowhere else you can find with that genotype and with that phenotype. Now we can see another one. This is capital R, small r, pink plus heterozygous dominant. Heterozygous dominant. Now we can count how many plants or sorry, how many organisms like this. Now here another one, pink plus hetero dominant. Again see here, pink plus hetero dominant, and here pink plus hetero dominant. Now with this phenotype, total how many are there? What is the actually here genotype? Capital R, small r, capital L, small l. Now this is pink plus heterozygous dominant, total 4. Total 4. Now we can see this genotype, capital R, small l, small l. This is red plus homozygous recessive lethality. Now how many will be there like that? Red homozygous recessive. Now we'll write here capital R and homozygous recessive, small l. Now this is red plus homozygous recessive, only 1. Now we will see this. This is a pink plus homozygous recessive. How many will be there like that? Pink homozygous recessive. You can say here pink homozygous recessive. Now here 1 and here 2. So we'll write the genotype capital R, small l, small l. Now this actually gives pink plus homozygous recessive. How many are there? Total 2. Right. Now we will see another one, this genotype. This is actually white plus heterozygous dominant. White plus heterozygous dominant. Let's see what is the genotype here. Small r, and capital L, small l. Now this is actually white plus heterozygous dominant. Now how many will be there in this phenotype? Let's see here 1, and this also same, white plus heterozygous dominant, total 2. And the last one, if you see, this is white plus homozygous recessive. This is small r, small l. White plus homozygous recessive. Now how many are there? Right? Only. So we can compare with the options. Now what is the ratio given here? 2:4:1:2, 2:1 we have got. Just for the ratio where we are going to match, we can change this according to that. 2:4:2, 2:1:2, 2:1. Now we can get the answer by seeing all the 4 options. Now see, the option is here, option 2. This is the right option for the given question. Once if you see here, this 4, wherever we are getting homozygous dominant and else, all the 4 will be valid. This and this and this. This 4 will be dying, and remaining 12 we will get the different types of phenotypes. Now the ratio 2:4:2:1:1:2 and 1. Option 2, the right in this question.